to welcome Professor Sanjeeva Srivastav. Professor Sanjeeva Srivastav is a scientist and group leader of Proteomics Laboratory at IIT Bombay. He is a visiting scientist in Arizona and Seattle, USA, and also a visiting faculty in China. He obtained his PhD from the University of Alberta, Canada, and postdoc from the Harvard Medical School, USA. Current research in his group centers on biomarker and drug target discovery and deciphering the protein interaction network in brain tumors using high throughput proteomics. He has published over 100 international papers, including Nature journals. He is a recipient of several awards, including the National Young Scientist Award Canada, Young Scientist Award Department of Atomic Energy and Board of Research in Nuclear Science India and the Department of Science and Technology India, and the Apple Research Technology Support Award UK. He has also received Best Teacher Award from IIT Bombay in 2014, 2018 and 2020 as well. Recently, he has started Cancer Moonshot India project along with Tata Memorial Center and National Cancer Institute USA. With this initiative, India has now become 12th country as part of International Cancer Proteogenome Consortium. Interesting proteomics updates and training material relevant to his talk will be found on Proteomics IIT Bombay YouTube channel. Now, I take this opportunity of requesting Professor Sanjeeva Srivastav to present his slides to everyone. Over to you, sir. Thank you.
Very good morning, everyone. Thank you, Priyanka, for very kind introduction. Thank you, Dr. Gandhi, for the invitation, and Professor Anil and all the faculty members at CRNTS for driving this initiative, which is, I'm sure, very important for the outreach and visibility of the center. I'm also a very active user of CRNTS facility, so let me thank the entire CRNTS faculty and staff for providing the excellent services, and especially Dr. Mayuri Gandhi for her all the activities and initiatives in the mass spectrometry based setups and various type of protocol and troubleshooting activities. So with this, it's really great privilege to present some of our recent work in front of all of you. Let me share my slides, and first I will start with giving you some overview of how mass spectrometry can be used for proteomics based research. And then I'll give you some glimpse of our recent work on COVID-19 research. So just to give you the glimpse that what we are talking is really a complex field of proteomics, which is aiming to understand the entire compendium of proteins which are encoded by the genome. So now we have a good idea for all the genes which constitute the whole genome of human and other organism. But if you think about from the genes, the transcripts are being formed, and from the transcript, the proteins are being formed. So from the same gene, you can have different functional RNA, and from there, you can have various type of modified proteins. So what we are looking at is really a complex sphere where we have very dynamic nature of the molecules, which are getting spliced, they are having various type of isoforms, and also they are showing post-translation modifications. So it's really difficult to capture this kind of dynamic proteome, and that's where various type of technologies are required. Mass spectrometry has definitely become one of the major workhorses in this area. So while genomics was accomplished around 2002 and 2003, when the draft human genomes were published, proteomics field is slightly, following at the same pace, but almost 14-15 years later only, first time we are able to now provide evidence that the gene coding proteins, they actually exist, and that was possible only because of advancement with the mass spectrometry. So the different researchers from different institutes, especially from Dr. Bernard Kruster's group from Germany and Dr. Akhilesh Pandey's group from Johns Hopkins, they first time showed that almost 17,000 human proteins for which we have evidence at the mass spec level that these peptides and proteins, they do exist, because so far it was only prediction. And this thing only happened in 2014 which was the Nature cover page. And next year to that, there was a group from Sweden, Dr. Matthias Ulhen's group. They published the tissue-based proteome map, which was based on the antibody-based approach. So as you can see that the field of proteomics is not very old. A lot of recent advances are happening, and much more activities are happening, in fact, in the last 5 years or so. So to govern, to accelerate, to organize the activities for the studying this complex proteome, and especially human proteome, there is an organization known as Human Proteome Organization, or HUPO, where we have a flagship project of Human Proteome Project, HPP. HPP has various verticals to investigate the human proteome problem, and mass spectrometry is one of the very strong pillars. We have also pathology pillar, antibody pillars, and knowledge-based bioinformatics pillars. All of these are equally important to trying to investigate the complex proteome and to really understand how the proteome is involved in driving various type of disease and biological questions. There are different teams which is formed as a part of the biology and disease-driven human proteome project. And also different, each chromosome wise, the task was divided to different countries to show that, which proteins belong to which chromosome and what are their roles. So again, you can understand that the field of proteomics, given the nature of the complex and dynamic proteome, is really requires collaborative efforts and really international effort of deciphering these kind of complex molecules. When it comes to mass spectrometry, what we are looking at the peptide-based analysis. So you have the complex protein mixture which you are digesting using enzyme like trypsin. Those form the peptides, and these peptides are the one which you are ionizing in the electrospray ionization, followed by separation using different type of mass analyzers. So you will generate a lot of peptides which can be detected using mass spectrometer. Now the search becomes very important, like what can be search algorithms which can be used to identify your mass spec data. And that's where the Human Proteome Project and HUPO provides guidelines to look into the very stringent data analysis criteria. So for instance, when you obtain the several thousands to million of peptides obtained from any type of mass spec experiment, for the search filtering criteria, you should have at least 2 unique peptides for a given protein. Then only you can be sure that what you're looking at is one unique protein. And these peptides should be not exactly to the adjacent region, but they should be non-nested, and at least 9 amino acid long length should be there. So these are some of the criteria and guidelines. Some of the viewers who are watching this presentation, if you are a mass spectrometry user and intended to publish your work in this field, I think it's important for you to follow some of these guidelines, which is continuously evolving. And now we have very stringent mass spectrometry data analysis guidelines. So I would ask you to refer to some of these guidelines. And only last year, based on these high stringency guidelines of mass spectrometry based data analysis, as a part of the HUPO consortium, international community, including our group, we published this work in Nature Communications. Now where we have idea for the human proteome greater than 90% of the proteome. And again, what was published in 2014 was still looking at almost 70% proteome. Now what we have idea after 2020 is 90.5% of the human proteome. It means for these proteins, the peptides exist, and we have evidence with the high stringent filtering criteria using mass spectrometry based data. And what you see here is kind of very complex data set, but it just shows you that there are different level of protein evidences which are existing. If we have the PE1 level evidence, which is highest stringency criteria, which I just described to you, those are almost 90.5% what we have. Additionally, there are some protein for which there is a prediction based on the RNA expression as well as antibody based test, and sometimes we have some putative information. All of those is at different level of PE2 to PE5. So please do not feel afraid with all the technicalities here, but only point so far I want to emphasize that investigating proteome at generating data level is not very difficult. but analyzing data is definitely very, requires a lot of your updates and your ideas and knowledge for these softwares and the filtering criteria. So to really decipher and decode the human proteome and various type of other life science system, different proteomic technologies have come forward, which have really made this task much easier. There are many technologies, but for this particular talk, I'm going to focus and restrict myself to only mass spectrometry based proteomics. But just to give you the flavor that proteomics is not only about mass spec, there could be different approaches of investigating the complex proteome. When you really want to understand what the proteins are doing in the body inside any kind of life science system, many times the questions are in a given response to a disease or response to a stress condition, or you're giving any kind of drug treatment, what are the proteins which are getting changed? And you want to analyze that at the proteome level, looking at all the possible proteins of that system. So one could use conventional gel-based approaches, including two-dimensional electrophoresis or dye technologies, followed by mass spectrometry, or you can also use directly mass spectrometry itself. Now, many times for specialized applications, people also use protein microarrays, and for studying the protein function and looking at their binding kinetics and drug interaction, one could also use the surface plasmon resonance and other type of biosensors. So there are a variety of technologies which are being used in the field of proteomics, from discovery as well as functional proteomics approach. But I'm going to restrict myself only on the mass spectrometry based proteomics. So as you're aware that mass spectrometry is very sensitive balance to say, where you are looking at the ionized species and measuring their mass and charge ratio, how well they could be separated in the magnetic and electric fields. So for any mass spectrometer, you have an ionization source, you have mass analyzer and detector. Now to start with your sample, if you are using ESI based MS platform, which is what we have in the Orbitrap systems, then you are going to interface that with the liquid chromatography. So we usually use nano LC, nano liquid chromatography, followed by electrospray ionization, and then Orbitrap mass analyzer. In fact, mass analyzers could be several type of mass analyzers, and depending on your applications, you can choose which type of mass analyzers you want to use. So shotgun proteomics is one of the more recent concepts. Maybe I would say more advances happened last 10 years or so, although this concept was given in 1998. Idea is, rather than separating the proteins on the gel, can we take the protein extract and digest that entire extract? During the digestion process, you are going to use the do the reduction as well as alkylation, reduction using DTT and alkylation using idostamide, followed by digestion of the proteins using trypsin, chymotrypsin, lyc, or many other enzymes you can use. Once those overnight incubation and digestion has happened, now these peptides, which is also having the buffer from your previous steps, those has to be cleaned. And now you can pass those from the reverse phase, these small columns, and clean up the samples. Then you can inject them for the MS1 or MS analysis, followed by data analysis. So this is in general any type of shotgun proteomics workflow, which is not involving any type of gel-based approach. It is directly taking the protein in the solution form and doing in solution digestion, followed by the mass spectrometry analysis. So as I was referring you to that, in the mass spectrometry based proteomics, you have the ionization source, you have the mass analyzers and detectors. So mass analyzer is very crucial because you are getting thousands to millions of ions inside the mass spectrometer, and you want them to better resolve so that these ions could now generate those peptide spectra, which can be utilized for proteome search. So different type of configurations are there, and at CRNTS you also have the 6550 series of Agilent, which is the QTOF mass spectrometer, where we have the quadruples and time of flight. So two different mass analyzers property are being used. Here, the separation is going to happen in the quadruples in the magnetic and electric field, followed by how much time these ions take to fly inside the TOF tube. So one could use these kind of hybrid mass spectrometer, QTOFs. But more recently, there is advent of these Orbitrap technologies, You already have probably dedicated talks on the Orbitrap technologies. I'm not going into technicalities of it, but what we are using currently is Orbitrap Fusion, which is one of the tribrid mass spectrometer. So here what you are looking at, we have three different type of mass analyzers from quadrupoles, Orbitraps, and ion traps. So again, for finite separation and at the MSMS level of analysis, if you have different mass analyzers, more than one for sure, you need hybrid technology or tribrid technology. It also provides you different options for looking at not only peptide sequencing, but also sometime post-transition modification and various type of specialized applications using these type of configurations. So this is of course one of the highest resolution mass spec currently being used for the nano LC, ESI, MSMS analysis. So based on these premises, I hope you have some idea now that the field of mass spectrometry, how it has evolved, and it is really crucial now to separate the complex mixture of any of these samples. And what I'm going to talk about is sometime, in our research we use the tissue proteome, but today I'll mainly talk about the swab proteomics and the plasma proteomics. Again, all of these are very complex samples. So when it comes to comparing the samples from your healthy individual to diseased individuals, now you want to do quantitative proteomics. So what I earlier told you was just the shotgun proteomics, you want to identify the proteins from the complex samples. But now you want to quantify all the peptides from the healthy individuals and all the peptides from the diseased individual, and you want to know which of these peptides or in turn the proteins are getting upregulated or downregulated. So you have various options available for doing quantitative proteomics. One approach is to look at the label-based approaches, which is either tandem mass tag or isobar tag for relative and absolute quantification, known as iTrack. After you have done the peptide digestion, those peptides could now be labeled at the N-terminal level with the TMT tags or iTrack tags, which binds peptide at the N-terminal sequence. In this case, what I'm showing you, let's say you have 3 healthy individuals, 3 diseased individuals, and you want to compare their differential proteome analysis in the same experiment. So you're labeling them with different type of TMT tags. And after labeling them, now you can mix all of these samples, all the peptides, pull them together, and then separate them from the nano LC followed by MSMS analysis. At the MSMS level, you will see these reporter ions, which is generating these fragmentation pattern for these reporter ions for TMT or iTrack tag. And this can be used for quantification. Now, there is a newer approach which is looking at without labeling also can we quantify the proteins, and that is known as label-free quantification. So here the same peptide which you have already prepared, rather than pooling the samples from different conditions, here you are going to run every sample independently, and you are theoretically generating the peptide library of each individual, which can be stored separately. Once you have done the same experiment, let's say for the same 3 control and 3 of the disease individual, now you want to look at what happens to the spectral count of these peptides at different retention time. So because you're in the liquid chromatography, you are separating the peptides based on their hydrophobic, hydrophilic properties. So if in a given retention time, if the healthy individual shows some level of abundance of the peptides and the disease individual showed higher level of abundance of that peptide, so then you should be able to quantify that by measuring the spectral counts. So this is something which is more newer concept, looks at label-free level of quantification. And much more recent advancement in that area is also known as DIA or data independent acquisition. Here, rather than looking at the entire M by Z region at the same time, we are looking at each of the small window to generate the high resolution one by one. So for instance, 400 to 500 M by Z, you generate a spectra for that, and likewise 500 to 600 and 900 to 1000. You generate these high resolution spectra, prepare that library, and then run your samples, and then now try to overlay that with the high resolution spectra to generate these analysis. So this is something which can give you much higher coverage. And our experience is that when we have used the brain samples, especially for brain tumor projects, with the label-free quantification level, we are able to get almost more than 3000 protein with high stringency criteria in single shot separation. Whereas in DIA, one could go even more than 4000 proteins. So that definitely provides much higher coverage of the proteome. So once you have identified the interesting proteins using your quantitative proteomics based workflows, then you can move on to validate those proteins. And validation has been quite challenging in the entire field because for validating your proteins, you need independent tests, not from the same mass spectrometer where you have acquired the data. You want to now test out your protein potential biomarkers or candidate proteins using different technology and more number of samples. So that's where triple quadrupole based mass spectrometers have become a very alternative resource for doing the validation of the proteins. Otherwise, we had no other way than to look for antibody based validation using ELISA or as well as various type of Western blot or immunoassays. So I'm not going in technicalities of how target proteomics are being performed. I may show you some slides as I go along in my data slides. But here aim is to look at specific targets. In the first experiment, when we wanted to look for the potential biomarkers, quantitative proteomics, we are interested to identify every protein and their quantification. After though that experiment is successfully done, now we have idea that which of the proteins and their peptide sequences are showing the differential abundance. So now we want to only zoom in and focus into those peptide sequences and want to validate those using selected reaction monitoring assays. And that's where triple quadrupole based mass spectrometer can selectively look for those peptides and generate their MSMS fragmentation, which can be utilized to measure those intensities and their quantification as well. So that I will talk in some time, but I will just show you very briefly that the field of proteomics, how it has evolved and some of the key milestones in this area in this documentary which we prepared for UPO recently. Alright, so proteomics is definitely much more newer addition of the omics technology as compared to genomics, which is much more advanced. But the field actually started in 1970s or so, 1975 to say, when two different scientists, Close and Farrell, they first time showed that the proteins could be separated based on their molecular weight as well as their isoelectric point properties. And with more and more advancement in the sequencing technology, in the mass spectrometry based technologies, especially ionization sources like electrospray ionization and MALDI, and different type of mass analyzers, including Orbitraps, the entire field of proteomics has really evolved over the years. The term proteome came in 1995 with Mark Wilkinson deciphering the term proteome first time. And immediately after completion of the Human Genome Project, the field of proteomics, really got evolved in 2003 or so. And now there are, very serious efforts by the Human Proteome Organization and the proteomics community to really trying to understand all the proteins of human and other type of model systems. So as you have seen previously, like what I talked to you, that there are more recent initiatives and efforts to really map all the proteins of human. And 2014 and 15, we first time got the glimpse. And only last year, we were able to get 90% or more coverage of the human proteome. So field is still open and a lot of challenges are involved. Especially people can also look at mapping those remaining 10% protein, which is known as the missing proteome per se. So while I'm sure this documentary, when you go to HUPO and you go to our videos, you can find more finer details of each of these discoveries, what has happened. I hope you are convinced that there has been a lot of efforts in the recent years, which has really made a robust mass spectrometry technology, and the genesis of that goes from 1950s or so, and only over the years, with the effort of the whole community, now we have such an ease of looking at the complex proteome mixtures with such an ease and looking at in a very high throughput manner. Now, there are studies, especially with Matthias Mann, who have shown that in 2 hours' time, you can separate the whole of the yeast proteome. So think about, without separating the complex proteome mixtures, now we have ability to separate the complex samples in 2 or 3 hours of gradient and obtain all their proteins in a very, very short time manner. So these are only possible with the efforts of the entire scientific community, and we are very excited and privileged to be working in this particular field. Let me now switch gears and bring you to the talk for which most likely you are excited about, that how mass spectrometry based technology, mass spectrometry based proteomics could be utilized to really trying to understand this invisible enemy, which has really last 1 year had made the turmoil in the whole world, and all of us got affected in different ways. So let's see, like, in which way our proteomic technologies and mass spec can try to understand this problem slightly better. So there are different omic technologies overall, which have really led to different ways of understanding this virus. And the RT-PCR test which came required the sequences, and that was not possible without doing the next generation sequencing to start with. But very quickly after doing the NGS sequencing from the Wuhan strains, immediately RT-PCR tests were developed. Also, the cryo-electron microscopy revealed the structure of these viral proteins, followed by various type of protein arrays and mass spectrometry based approaches. New targets were identified, and different therapeutic strategies were used to combat this problem. So again, in a very short time, the scientific community really tried to use all their arsenals to understand this virus COVID-19 better, SARS-CoV-2 virus better, and understand this disease COVID-19 and try to contribute in a more meaningful way. So what we thought to first look at the primary sample of COVID-19 patients, which is nasopharyngeal swab. And for this work, we did collaboration from different hospitals of Mumbai, especially Kasturba Hospital and Noir Hospital, also Jaslok Hospital. But Kasturba Hospital, most of the samples came from, where the patients who were coming, their nasopharyngeal swab was collected. And this same sample was used for the real-time PCR test for detecting the viral load. Now, can we detect the peptides of these virus SARS-CoV-2 from the nasopharyngeal swab sample? And that was our first, kind of curiosity, as well as our challenge to look at how to, identify these peptides from these nasopharyngeal swab samples. We have never worked on the NP swab or oropharyngeal swab samples in the past. And if you know that, when people are being tested for COVID with the real-time PCR test, their swab is, being collected, which is put in the VTM tube, which is the viral transport medium tube. And this VTM contains some abundant proteins, including albumin, as well as various other, sugars. All this is then, being preserved in the cold condition so that the RNA can be extracted easily, because then that can avoid the transport issues and the storage issues. So the VTM is actually going to be, which is very much required for the RNA-based work, but that actually an interference for the proteomics work. So that was one of the challenge, like, how to overcome the medium in which the samples are being collected. And also the safe handling of the sample is very challenged, because how to handle these samples, with utmost precaution and safety. So with some of these challenges, our questions which we thought to address, how to develop these SOPs for viral peptide extraction from these nasopharyngeal swab samples. Also, how to look at the deep proteome profile of these patients, both of their viral peptides as well as the host proteins. So given that these are respiratory specimens which are coming, we are hoping that there will be viral peptides and viral proteins present in those samples, but also because it is coming from the human samples, human nose samples, so that will also contain the respiratory specimen of the human protein and host protein origin. So while you are extracting the proteins, it should have the mixture of both host peptides as well as the viral peptides, which can be deprofiled using orbitrary fusion. And also, once we know which peptides are coming more in the common pattern in the diseased population, can we develop the targeted assays based on the MRM based work using ARTIS triple quadruple mass spec. So these were some of the key considerations which we had for nasopharyngeal swab samples. So the workflow, as I described, that these samples are collected in the VTM tube with the nasopharyngeal swab, RNA being extracted and cDNA prepared, sample being taken for the real-time PCR-based test, where you are looking at the CT values to look at the viral load and whether the patient is positive or negative. Now, same VTM tube in the hospital itself, we provided our staff over there who can prepare the sample and immediately precipitate out these proteins in the various organic solvents. And again, we chose these organic solvents so that the viral inactivation can happen very quickly right away after the sample collection in the BSL-2 labs there. So then ethanol, isopropanol or acetone, all three of them were tried. And then a mixture of those was used for the protein digestion, followed by the peptide estimation and separating them in the fusion to look at the deep proteome or the global proteome analysis, as well as eventually we refined the peptide list and developed these kind of selected reaction monitoring tests. So as I mentioned initially, we had no clues about, given the interference of the VTM tube, that how we are going to get the peptides from the virus, what kind of methods are going to work better. And we have to also look into all the safety considerations of the sample preparation. So when we prepared samples in isopropanol, ethanol or acetone based methods, these are the number of unique peptides which we could detect from the viral origin. And so we did find a lot of viral based peptides which are coming in these samples. But what we thought that maybe the pool of all these extraction methods of organic solvents could provide us enriched of peptides from all of these various extraction methods of these samples. What you see here is the chromatogram of the samples from the NP swab, and which represents both the host as well as viral peptides. Now for search, database search becomes very crucial here. So the same set of data, you can first search for the SARS-CoV-2 database, and then the same data was used for the human proteome database search. So we were able to identify 4 peptides of spike proteins, 3 of nucleoproteins, and 4 peptides of replicase proteins. And these 11 peptides were then further selected to develop the multiple reaction monitoring test to find out, can we see these peptides in the COVID negative versus COVID positive patients, and within COVID positive patient as well, if there is the low CT value or the high CT values, whether we can see the difference. So it means if we are looking at the viral load, is there any correlation with the peptides, what we observe here in the sample as well. So this is more technical data, but it just shows you the profile of multiple reaction monitoring based test, where as compared to the negative and the the positive patient, we could see differently the change in the intensity of these peptides. And also we could see that in the low CT value, which is showing you high viral load, that's where we have higher peaks, and the high CT value, which is the low viral load, is showing you the low peaks of these peptides. So while doing this work, what we realized that there are definitely good opportunities of doing mass spectrometry based methods for doing viral detection. The extraction methods are quite simple because we are simply using the ethanol, isopropanol or acetone just to to precipitate these pellets and then reconstitute those in urea buffer, followed by mass spec analysis. Now for RNA based work, the major challenge becomes the storage and handling of these samples and preserving those in the cold condition, so which could be overcome with these kind of simple extraction methods. And of course, mass spec is much more sensitive as compared to real-time PCR or any other test. But there are many challenges as well. It was, of course, our team was very well trained for doing this kind of tests and developing these kind of assays. So we were able to do that in a very short time. But to really employ this kind of methodology at the clinical setting is not easy, given the challenges of the instrument procurements and the cost of instruments, the specialized skilled manpower required for operating these kind of tests. And of course, the throughput becomes very challenging as well, because in mass spec you can run only one sample at a time. And then the sample preparation requires overnight steps and a lot of methods of SOP preparation. So all of that makes it slightly challenging to really make it easy deployment for the hospitals. But nevertheless, it did show that mass spectrometry based platforms can be used to do the SARS-CoV-2 detection. And not only our own work, but while we were doing this work, some other publication also came forward from different groups, which showed that different mass spec and different groups are able to now develop the test for doing SARS-CoV-2 detection using mass spectrometry. But one thing which we found more interesting was essentially looking at the host response, because that was quite surprising to us that how many proteins we will get from the nasopharyngeal swab sample, which is of the human origin or the host origin. But after doing this kind of test, we could actually find almost 1200 plus proteins were seen in all the samples, which was a decent coverage. And then throughout the experiment of any mass spectrometry, you have to run some quality control checks, which throughout in the large number of these samples run, we were running the QC pools, which showed very good correlation, as you can see on the right hand side. And then when we wanted to compare the COVID-19 negative and COVID-19 positive patients, we could see that the signature of the proteins which are quite different in both of this group. So it was easy to distinguish even from looking at the host protein response, the COVID positive and negative patients. And some of these proteins which are shown here on the screen are the ones which really showed good upregulation of the in the host of the COVID-19. And some of these proteins are also being tested in the clinical settings right now, but we're using the routine test of lactate dehydrogenase and other kind of tests which are being done, which is also indirectly connecting that these proteins what we see are really relevant in the clinical test as well. We also looked at whether within the COVID positive patients can we look at the protein signatures of the non-severe and severe patients. But here the segregation was not so great. We could not see that level of segregation what we could see in the COVID positive and negative patients. But overall, what we could identify that the various pathways which are changing in the host in the COVID-19 patient because of the infection from the SARS-CoV-2 virus, they are majorly belonging to neutrophil degranulation pathways. Various type of platelet activation and degranulation is involved, and protein machinery for the mRNA translation is also heavily perturbed. So some of these key proteins and pathways are the majorly modulated in these COVID-19 patients. So looking at the host samples, what we realized that it is possible to even look at the host protein response and use that for disease severity monitoring. And that could actually have more immediate clinical requirements, given that RT-PCR is already being used for the testing a patient, that patient is positive or negative, but what is going to be the patient response for the disease in terms of the patient will remain mild or it might progress towards more severity. And that's where the host proteins can be much more powerful and provides more opportunity for clinical testing. Challenges for doing this type of test remains like the limitation which I discussed earlier of the instrument cost and the specialized manpower. We also looked at the COVID-19 patients' plasma samples, which are again COVID negative as well as COVID positive, non-severe and severe group of patients. We have good know-how of doing the proteomics of infectious disease from the blood samples. Very recently, we had published one paper in Communication Biology on malaria, where a similar kind of problem was investigated to look at the patients of non-severe malaria and severe malaria, and how this type of plasma-based proteomics can be utilized to find out the potential biomarkers and the mechanistic insights of the disease. So our team was very much tuned for doing this kind of work. We had all the reagents available for doing this experiment. In the lockdown time, of course, we were also constrained with the reagents and how many samples we can analyze. But fortunately for this experiment, we had everything in-house available to do this experiment. So when we got these patient samples of COVID-19 negative, COVID-19 non-severe and COVID-19 severe patients, first challenge is to prepare these samples where the plasma contains many abundant proteins like albumin and IgGs, which has to be removed, and you have to deplete those using various type of antibody-based approach. So these depletion columns that becomes very important and cost limiting factor as well for sample preparation. So we depleted all these plasma sample individually for each patient, prepared the peptides, separated those peptides using Orbitrap technology. And once we finished the data analysis of various peptide and protein based investigations, then we took the selected peptides and took them to different mass spec triple quad artists and looked for the MRM based validation and followed by the pathways and network analysis and potential drug repurposing aspects. So the host proteins actually reflect the disease quite well, because many proteins which are leached out from different systems, they come to plasma, and then plasma is a very accurate reflection of what is happening in the host response in any given disease. So in the COVID-19 also, more than the swab sample, we were able to see the host response are really reflecting the good disease biology. And here you can see the good segregation of COVID-19 negative and positive patients using some of these proteins. But we were also more keen in fact on looking at non-severe versus severe population, whether we can make the prediction that how a non-severe or mild patient can progress toward the severe type of disease of COVID-19, because the limitation of the ICU beds and the treatment modalities could be then minimized in that manner. So we were able to find the distinct signature of the proteins, here much more profound using the plasma samples. And we thought, can we take some of these leads forward for the clinical translation value? And that's where first we looked into various pathways where these proteins belong to. And of course, blood coagulation and complement cascade pathways came quite abundant. But also we see certain things like glucocorticoids and various type of exocytosis-based mechanism. We are not very sure is that these pathways are coming because of the drug response, the drugs which have been given to COVID-19 severe patient, or is that the real, clues itself. Those things are still very difficult to really investigate, given all the patients are already undergoing some treatment. So you have to take those bloods, and you have to keep that in mind as well, that the responses what you see for different pathways, it might be in response to given drug as well. So nevertheless, we could see very distinct response of various protein, and we selected some of these proteins shown here, for which we developed the test of multiple reaction monitoring. And you can see very clear distinct peaks here of non-severe and severe patients, where we are looking at set of these proteins belonging to the serpin family. And these serpin proteins, which are various serine proteases, they are quite showing abundance in the severe patients. Apolipo B protein, as well as various type of proteins of angiotensinogen, AGT protein here, and the fibrinogen gamma chain proteins. Many of these proteins actually showed very high response in the COVID-19 severe patients as compared to non-severe patients. And this is very exciting for us, that we are able to make now some prediction of the disease severity using the host blood sample, which is currently not possible from any type of test. And that's where I think mass spectrometry can serve much more utility of providing some novel insights of the COVID-19 patients. We also did some database search based on the proteomics data, the proteome data, what was available to us, to look at the human protein and the viral peptide interactions to develop the interactome map. And finally, after identifying the set of protein, which I already discussed briefly, like various serpin proteins and ascended A9 protein, we looked at various type of FDA approved drugs and did various type of docking studies to find which of the drugs can be used for the repurposing drugs, which can bind to maximum number of these proteins which are involved in the disease severity. So this work is still in progress and needs much more validation before it can be taken for any kind of drug discovery work. But it definitely showed good promise that this drug selenoxir can actually inhibit all these three proteins, which is showing very high upregulation in the COVID-19 severe patients. And likewise, ponatinib showed against these two proteins of serpin family. So again, some of these drugs could be utilized as a drug repurposing approach, but it will require more validation. But given that these are already FDA approved drugs, I think their translation could be much more smoother as compared to looking for the new drugs. So overall, I think we have been quite busy in the in these months of investigating COVID-19 patients. There are series of activities, series of experiments are being performed. Something I spoke to you was essentially on the global proteomics part. I have not spoken to you about some more initiative on the metabolomics work, as well as some of the work on the FTIR based investigation, and also more recently about microarrays and some of the NGS based approaches to investigate these problems. So to summarize my talk, we have identified set of proteins and potential peptides which could be developed for these type of selected target assays, looking at selected reaction monitoring. Peptide detection from SARS-CoV-2 virus is possible, but given the challenges of developing the cost-effective test and clinically deployable test, it may not be best approach of taking forward. Nevertheless, the host response proteins, they are quite promising and they do indicate the disease severity progression, which is not possible so far from any approach. So mass spectrometry can play an important role. And these proteins could also be taken forward for developing ELISA-based test or more clinically friendly test in the future as well. So based on this work, we have already filed a couple of patents for the SARS-CoV-2 detection and the disease prognosis. And one paper on the swab proteomics is just provisionally got accepted in iScience. And another work on the plasma-based proteomics is under review currently. And there are more work which is being communicated. So there has been, large number of, team members who have been involved, few physically, more virtually, but they have all been actively involved. This is a swab team, this is the plasma team, the drug team, the multiple election monitoring based validation team, FTIR investigation, plasma proteome metabolomics team, various students who really worked in the hospitals and the BSL-2 labs, clinical team, and different clinicians, Dr. Jayanti Shastri, Dr. Sachi, Dr. Om Srivastava, Dr. Michelle Hill from Australia, and various UGs and interns. So a large number of students and collaborators have really helped us to contribute something in this particular area. Of course, it was not possible without generous funding from the various funding agencies, from the SERB, DST. We were actually fortunate to have one of the first funding from the SERB, DST, Department of Biotechnology, Indo-US Science Technology Forum, and seed grant from IRCC for COVID-19 projects. Various industries have also been very proactive and supportive. Thermo Fisher Scientific, they did all possible support for mass spectrometry-based workflows. Agilent was very helpful for the FTIR work. There are two more industries which are coming forward to help us to do more translational values of these products and these projects. So before I close, let me show you a 1-minute clip of an upcoming workshop which we are going to conduct at IIT Bombay. It will be essentially more online mode, but you may have some opportunity to participate. So let me play this clip. All right, so please keep an eye on this workshop. We'll be happy to have your participation in this online activity. Also, we are uploading a lot of study contents and various type of tutorials on our YouTube channel of Proteomics IIT Bombay, where you can listen a lot of lectures which we deliver for IIT Bombay courses, as well as different other workshop hands-on activities. So please make use of these resources and a lot of training which you can obtain virtually. And of course, we will be very happy to have you in person discussing some of these things in our lab here. We have also published several special issues on proteomics. In the free time, please do read these and ask us for the more editorial contents. And finally, I would like to say that the virtual mode of interaction is really helping us to continue our engagement despite the challenges of COVID-19. And there are various online courses which we offer, MOOC courses which we offer on proteomics, proteogenomics, and currently we are running bioengineering and interactomics courses. Some of these are really able to connect us with large number of students and participants from all over India and world despite this difficult time. So thank you so much for your attention, and I'll be very happy to take any questions. So Priyanka, I'm all done. So I'm open for
Yeah,
any
so
questions.
yeah. Thank you, Professor Sanjeeva Srivastav. Let's begin with today's question answer round. So the first question is anonymous. Tell us about the role of LCMS in the development of anti-COVID-19 drug, like example remdesivir.
If I, yeah, so I'm just looking at on the Q&A here as well. So tell us about the role of LCMS in the development of the anti-COVID-19 drug. Uh, See, I think, initial drug discovery, because given the short time what was there for looking at the COVID-19 patient treatment, it was not possible to really discover something very, new in a very short time. So that's where FDA approved antivirals and even anti-malarial and various type of known drugs were tried out, right? So aim was not immediately to discover something very new, but also to first try to repurpose what is already FDA approved, regulatory agencies which has already approved those type of drugs. So for instance, if you are going to use some drugs, they which are very new, which are identified from mass spec approaches, they may show some side effect. So that's where it becomes very crucial to try out something which is already approved and then play with that in the beginning. But over the time, the scientists actually tried out some immunoprecipitation based studies and looking at more interactome analysis of looking at how SARS-CoV-2 virus in the cell cultures of the cell lines, that interacts with the host proteins and followed by different type of drugs which could be utilized. So almost a list of 60 or so drugs were prepared based on these kind of interactome analysis. And again, many of these things cannot be just done right away at the experimental level, but a mixture of experimental evidence using the immunoprecipitation interactome analysis of the IPMS based work, followed by computational predictions of these potential drugs which are binding over there, has really helped us to evolve the field and try to find out, which could be potential drugs. But the drug discovery process and the drug deployment is quite tedious and challenging, as you know. So therefore, it's not only the mass spec immediately which could do that. A lot of antiviral assays has to be done, various type of knowledge which the virologist already had in the field, and different type of drugs which are already being used. Those all together has really contributed for some sort of treatments for remdesivir as well as other type of drug. But still, there is a lot of scope for looking at more specific treatments and in different population groups. For example, chloroquine did not work out so well as it was anticipated. But given that not what we see is there is some very specific response which is coming from the non-severe to the severe patients of the COVID-19. And there are recently, or at least 4 or 5 papers have already published, very big journals, which is also supporting our findings, like what we have already communicated but has not yet published, but already some papers have already come in the preprint, which is showing that the similar set of proteins are actually in different populations of the world are showing the lot of changes. So there is a good way now to, we have the evidences that the patient derived proteins, that data from the mass spec, that can we target some of these proteins and look for more effective therapeutic strategies against these. So now there is much more scientific process and evidence involved of developing the drug in the long term. But of course, what was being tried was on a very short-term basis, urgent basis, with available knowledge from different coordinates. It was not alone proteomics or mass spec which could have contributed just for the COVID-19 patient treatment.
So actually, only one question we got. There are no more questions. So
all right,
we are really great.
Is quite new. I'm sure
Yeah.
People may not have so much idea for this, but
Yes.
I must say that this was really exciting and challenging project for us. But given the power of these mass spectrometers and orbitraps, I think it was really possible to really decipher this complex proteome on a very short time, and that really helped us to find some important clues, which are quite relevant now for these patients, and our clinicians and collaborators are very excited for taking some of these leads forward.
So one more we got recently. It is anonymous. Is the virus mutating as it spreads?
Can you repeat the question? I could not hear clearly.
Yeah. Is the virus mutating as it spreads?
Is the virus mutating as it spreads?
It spreads. Yeah.
Right. I may not be the best person to comment on this question because I'm neither epidemiologist nor the genomic scientist. Assumption is, as a general public, one would assume that these viruses, especially SARS family viruses, they do show extensive mutation. And if you look at some of the WHO data and genomics data that do refer that there is a lot of mutations happening in these SARS-CoV-2 strains currently, and also in India also we have various mutation. Only that it's one of the UK strain and South African strain which has really become much more lethal and shown very fast spread, which we have better idea. But nevertheless, there are many mutations are happening currently, and there is a really need to continuously understand this virus. The problem has not yet gone completely, so we have to really be cautious about looking at their mutation patterns, probably what proteins are getting changed because of those mutations, what type of symptoms are different in these patients, and what way one could really develop the more effective vaccination strategies in future. All of those are possible if we really continue our investigation using definitely NGS as well as mass spectrometry based approaches.
Okay, so we are really grateful, sir, for answering the questions. So on behalf of SEF CRNTS, I would like to thank Professor Sanjeeva Srivastav for interesting talk on MS-based proteomics for COVID-19 research. I would like to thank our head Professor Anil Kotantharil and our IT team members Mrs. Nidhi Goyal and Mr. Jagdeep Singh for the continuous support in this webinar series. And last but not the least, I thank all the participants for attending this webinar and making this event successful. A link for the feedback